Oh, thanks. Recording is started. Today we're gonna recap the homework assignment that we talked about last week, and there's a new homework assignment for this week. Don't worry, it's not that hard of a homework assignment, but I would like people to take this homework assignment. And wouldn't you know, the minute we start class, I'm getting like texted and called and all kinds of stuff, and it's been such a quiet morning. But okay, so and then we're going to go over a clinical case of hemophilia, which is such a common disease, but it's remarkable how much complexity there is in the genetics of hemophilia. And then we're going to spend the rest of the class talking about inheritance patterns, and we're going to introduce you to what a family tree looks like. And then we're going to talk about the typical kind of monogenic Mendelian inheritance patterns. And then we're going to look at all the variations, or the so-called non-Mendelian inheritance patterns. So any questions before we dig into the homework assignments? Okay. So this homework assignment, this is the same slide we looked at last week. Just as a reminder, I'd like people to begin prepping for participation in an ethics debate. This will be topic 12 coming up in the end of July, mid-July. So this is just a reminder. If people could begin kind of thinking, or, you know, even as you're stumbling across articles or as you're stumbling across emails, if you see emails or articles that are talking about ethical aspects of genetics, you know, give them a quick read, and let's try to make this topic 12 session, you know, a very active, open debate and participation amongst the class. It's not going to be me teaching anything. It's really going to be you guys debating. We have a new homework assignment this week, and I'd like to have all of this done by next week, and it should take you about 5-10 minutes to do this homework assignment. You can obviously spend more time on it if you're interested. Normally, what we've been doing over the last several sessions is when we look at a case, and we look at the bioinformatics aspects of the case, and then we look at the informatics aspects of the case, we've been focused on how do you communicate the variant information, how do you communicate the actual genetic results. But today, because the topic is on inheritance patterns, we're going to be focused on the representation of family history, structured family history. And there's a couple of different standards in HL7, at least 3 different ways of representing family history in HL7. One is the HL7 version 3 pedigree model. One is the representation of family history in CDA or through the CCD standard. And then there's a couple of FIRE resources for representing family history. One is the basic family history FIRE resource, and one is the extended genetic FIRE resource. We're not going to be looking at the CDA resource today, but we are going to be looking at the HL7 version 3 pedigree model, and we are going to be looking at the FIRE resources. This particular application, familyhistory.hhs.gov, is a graphical user interface that allows you to construct a family history, and then it saves the output as an XML file, where that XML file is aligned with the HL7 version 3 implementation guide. So what I'd like you to do is go to that page, create a family history. Just be sure to not include anything personal or private in these, because you're going to be sending them to me, and they might be something worth illustrating or worth sharing with the class. So if you're completely uncomfortable doing this exercise, just skip it. But I would encourage you to at least include something fake. So I'd like you to include at least grandparents, parents, siblings, and children in your family tree. And I'd like you to include at least one condition, which can be fake and which you don't mind making public. So you create your family history using the graphical user interface at this website. and save the data. And when you choose to save the data as an XML file, it downloads the XML file to your computer. And that XML file is based on the HL7 version 3 implementation guide. So take a look at it. Just inspect the XML file so you get a little bit familiar with how that file is structured and how that file is representing relationships to the patient and how that file is representing different diseases. Within the graphical user interface, you can also generate a pedigree drawing, and you can tell the pedigree drawing to highlight the condition. What I'd like you to do for full credit is send me your XML file and send me your pedigree drawing. I'll take a peek at them, and then you'll get full credit. Now, you might ask, what does that mean to get full credit? And that's a good question, but we'll cross that bridge next week, okay?
Does that mean I won't flunk the course, Bob?
Well, you're going to get, Dan, you're going to get something special for credit, okay. But it's yet to be determined what it is.
All right.
Okay. All right. So that's the homework assignment. I'd like to have it in by next week. All right. We're going to dig in with the case unless there's any questions. All right. So today we're going to be talking about hemophilia, and this is a kid with hemophilia, and the kid bonked his head, and he's got this huge contusion on his forehead, and it actually took about 6 months for this contusion to resolve. The case is a patient named ST, and I'll tell you her name is ST only because when I show you later the family tree, you'll see I went into that family history tool and I created ST's pedigree, which I'll show you. And by the way, the pedigree for this case in HL7 version 3 and the pedigree for this case we're about to go over in FIRE are also available at this website. So we'll be going over a couple of pedigrees, and all of those are available at the same website. So ST is a healthy 38-year-old woman, and she went in for counseling to check on her risk for having a child with hemophilia. She has a maternal uncle, so her mother's brother died in childhood from hemophilia, and she has a brother with hemophilia. No other family members have bleeding disorders, and the geneticist explained to ST that her family history was suggestive of an X-linked abnormality of coagulation, likely hemophilia. And so to confirm the diagnosis of hemophilia, the geneticist told ST that her brother should be evaluated first because, as we'll talk about, there's lots and lots, hundreds of different possible mutations in the factor 8 gene and the factor 9 gene that could be responsible for hemophilia. And oftentimes when you see a variant in a gene, it's hard to know whether or not it's clinically significant. So in this case, the geneticist told ST that rather than doing genetic testing directly on ST, it would be better, since her brother is known to have hemophilia, he should be evaluated first. They should try to figure out what mutation the brother has, and then they can test ST to see whether or not she has the same mutation. So ST talked to her brother. He agreed to the evaluation, and in fact, he was found to have a mutation, in this case, in the factor 9 gene, consistent with hemophilia B. They went and then tested ST and found that she did not carry the mutation. So they tested the brother. found that he had the mutation, and then went back and tested ST and found that she didn't. So this is a pedigree of ST. This is the patient, and you'll see the arrow. The arrow in a pedigree typically points to what's called the proband, or the person who's seeking guidance. And in this case, we don't know her status. We know that the brother, as indicated by the filled-in box, has the disease. We know that the maternal uncle died from hemophilia, so you see the slash across him. In this case, we are inferring that the mother is a carrier. The mother doesn't have any symptoms, but we're assuming that the mother is a carrier because the probability of brand new mutations causing hemophilia in the maternal uncle as well as the brother are pretty low, and it's far more plausible that the parent up here transmitted the gene to the uncle and to the mother, who then passed it on to the brother, and now the question is whether or not ST has the mutation. So we're presuming that the patient's mother is a carrier. A couple other pictures just showing some of the clinical manifestations of hemophilia, and you'll hear about hemophilia A and you'll hear about hemophilia B, and we'll talk a little bit about the distinction between those. Hemophilia A is a mutation of the factor VIII gene, and hemophilia B is a mutation of the factor IX gene, but clinically they're indistinguishable. They both result in severe bleeding disorders. Well, they can be very severe. They can be mild, moderate, or severe, and they can be life-threatening. A minor bruise can cause quite a huge hematoma, and recurrent bleeding into the joints can cause chronic joint damage as well. This is a pedigree from dating back to Queen Victoria. Ultimately, it was determined that Queen Victoria was a carrier for hemophilia B, and it's just kind of a nice illustration of a very complicated pedigree tracing the mutation pretty extensively through a family. Again, the question marks signify that the genetics aren't known in here. And I think we'll see here in this family tree some evidence of consanguinity, but it's such a confusing picture. Anyhow, so this is an example of a pedigree for hemophilia, ultimately determined to be due to mutation in the factor IX gene, resulting in hemophilia B. And, what discussion of hemophilia would be complete without the obligatory clotting cascade pathway, right? So the factor VIII gene is responsible for producing the factor VIII protein, and the factor IX gene is responsible for producing the factor IX protein. And it's not illustrated very well on this particular picture, but factor IX, when it's activated, is responsible for activating factor X. And factor VIII is a cofactor in the activation of factor X. So you really have factor IX and factor VIII working together to convert factor X from an inactive state to an active state. And once factor X is activated, it can continue the clotting cascade. It can activate prothrombin to thrombin, which can then activate fibrinogen to fibrin. So what happens here is inside a blood vessel where you have a nick or a cut, you initially have the platelets come to the region and they start to form a plug. But the platelet plug in and of itself is somewhat fragile. And the platelets become enmeshed in this fibrin network, and that helps stabilize the clot. And ultimately, once the actual source of the cut, once the exposed edges of the cut, if you will, are covered in platelets and fibrin, and there's no more exposed edges, then the triggers for platelet adhesion and the triggers for clotting are then turned off. And so that's what then self-limits the coagulation process so that it doesn't in and of itself become problematic. So what we have in hemophilia is a deficiency of factor 9 or a deficiency in factor 8, and as a result, we can't activate factor 10, so we can't ultimately form the fibrin meshwork to stabilize clots, and so you have slow oozing at the sites, resulting in these large hematomas. The genetics of the factor 8 gene are, I mean, they're just remarkable. They're just remarkable. And the mechanisms by which the mutations develop on the factor 8 gene are also pretty remarkable. So we're looking here at chromosome 28. This is, again, we're looking at the UC Santa Cruz genome browser. And if you're interested in looking at this in any more detail, the URL is in the slide deck. So this is actually what we're looking at right here. Okay. And here's the factor 8 gene. Okay, it goes from here to here. And again, this is another gene that goes in this direction. Okay, so remember, here's the chromosome, and at the bottom you can imagine a strand that goes from 5' to 3', and on the top you have the anti-parallel strand that goes from 5' here to 3' here. You can have genes on either strand. Now, all genes are read from the 5' to 3' direction, but you can have genes on this strand that would go from left to right, or you could have genes on the anti-parallel strand, and in this visualization, they would be transcribed from right to left. Okay, if that's fuzzy, let me know. But basically, all genes are transcribed 5' to 3'. But when you're looking at a chromosome, there's two strands. There's the 5' to 3' strand, and then there's the anti-parallel strand. And a gene can be on either one of those strands, and therefore it can be confusing when you look at a gene, because this particular gene here is transcribed. Here's the beginning of the gene, and it goes this way. But in the same view, you might find other genes that, for instance here, that start here and go this way. And if you zoom in on the browser, you can actually see which way the genes go, because you'll see little arrows on the genes. So here's the factor 8 gene. Here's the first exon here. Okay, and it goes this way, and there's 26 exons in this gene. The gene spans about 190,000 bases. And one of the things that's interesting about this gene is this is the first transcript, but this particular gene yields a second messenger RNA. So here's a gene that has the primary messenger RNA that we're familiar with, and that's what forms the typical factor 8 that we're familiar with, that we looked at in the previous clotting cascade slide. But we also see a second transcript, and that's why you'll see in this view, you'll see factor 8 twice, because there's actually two messenger RNAs that are known to come from this particular gene. This second transcript is not well understood. It's called a putative small protein. It's an isoform of the protein, and it's believed to play a role in clotting as well, although its function is not nearly as well understood as the primary transcript. When you look at this picture of the factor VIII gene, again, you'll see the lines here correspond to the exons, and then the little spaces in between correspond to the introns. So remember, during messenger RNA production, all of the introns are going to be spliced out. So all of these introns are going to be spliced out, and the actual messenger RNA will be all of these exons sandwiched together with the introns spliced out. Here, we're looking at, this is intron 22. And interesting, there is another gene on the anti-parallel strand called factor VIII-A1. Okay, so this is a gene that's contained entirely within intron 22 of the factor VIII gene, and it's read in the other direction. So on one strand, you have a gene that goes from left to right, and on the other strand, you have this little gene nestled in an intron of the factor VIII gene that goes in this direction. The function of this gene, the factor VIII-A1 gene, is actually not known, but we see this gene in other mammals. For instance, we see that it's active in the mouse, so it's presumed to have some function. And also, when they measure RNA transcripts in cell preparations, they see that this gene is transcribed abundantly in a wide variety of cell types. So the actual function of this gene isn't known, but the fact that it's also expressed in mice and the fact that the gene is widely expressed in a lot of human cells suggests that it's playing some role. We just have no idea at this point what its role is. The other thing that's interesting about this gene is that there's two other copies of it. So here's the F8A1 gene, but there's another copy here closer to the telomere of the Q arm of chromosome X, and downstream even further, even closer to the telomere, there's yet another copy of F8A1. So it's kind of a strange occurrence where we see this little gene here, and then we see it copied down here. And that actually is important for understanding what happens when the factor VIII gene gets mutated, and that's illustrated here. So there's hundreds of different mutations that will cause hemophilia A and hemophilia B, but it turns out that in patients with severe hemophilia A, roughly 40% of them have what's called an inversion mutation. And the inversion is believed to occur during meiosis, and it's believed to occur, now normally during meiosis, we talked about the fact that there is recombination. And during recombination, if you recall, for instance, chromosome 7 that you derived from mom will pair up with chromosome 7 that you got from dad. So chromosome 7 from mom and chromosome 7 from dad will pair up, and crossing over will occur. Pieces from one chromosome will switch over to join the other chromosome and vice versa. Well, in a male, there's only one chromosome X. There's only one X chromosome, and the X chromosome can line up and have crossing over intrachromosomally. It can cross over with itself. And so we have in intron 22, we have that F8A1 gene, but we know that near the telomere, we have two other copies of the F8A1 gene, and because of the homology between these two segments, they can line up and crossing over can take place. And if crossing over takes place and then the chromosome opens back up again, you wind up with this inverted segment. And so what we see, again, is in severe, and obviously this just completely destroys the gene. No protein is produced when you have this large inverted segment. It's a very severe mutation, and it's the mutation that's seen in 40% of people with severe hemophilia A. The other thing about this worth noting is that the spot where the crossing over takes place differs amongst families. And what makes it a little bit difficult in, as we'll talk about, is how do you then formally name this mutation so that you can search for this mutation in databases. The mutations we've been looking at before are generally they've been point mutations. They can be named very precisely. Here's a reference DNA sequence, and 500,023 base pairs into it, the reference DNA has an A, and in the variant we're looking at, it's a G. But here, the actual endpoints sometimes aren't known because older technology didn't allow for precise determination of exactly where the inversion endpoints were. Or what we also see is that the endpoints can differ amongst people. Now, this is all kind of gradually evolving. Actually, maybe I shouldn't say gradually, maybe I should say rapidly, because we used to study inversions using some of the older technology that we'll talk about when we talk about genetic testing. More and more, we're studying DNA using next-generation sequencing, where our ability to identify precisely where the endpoints are is continuing to improve pretty rapidly. So we're in this transition where when we talk about these large structural mutations like this inversion here, probably over the next several years we'll be able to refer to them precisely. But right now, our ability to refer to mutations like this using precise syntax is a little bit fuzzy, and I'll show you that when we begin talking about the bioinformatics aspects of this particular mutation. Let me just pause there because that's a mouthful. How are you guys doing? Okay so far?
Yep.
Okay. Is this perhaps one of the most interesting mutations you've ever seen?
I was just going to say that.
Well, yeah. I mean, isn't it crazy how this happens and how people have identified it, and the fact that there's all these copies of this little F8A1 gene, and even the fact that there's a gene within a gene. It's just, it's kind of hard to wrap your head around how all of this stuff, came to be. So, as I like to do with all of our clinical cases, I like to then dig in a little bit to the bioinformatics aspects and the informatics aspects. And again, I differentiate between bioinformatics and informatics. Bioinformatics is kind of like what the researchers do. It's all of the public databases and utilities for studying genetics and studying huge databases for research, whereas the informatics is more of the clinical HL7 SNOMED type stuff. How do we communicate this information? And admittedly, there's a blur between the two. So the first thing that I like to do, well, one of the use cases that I like to raise with all of these cases is, okay, let's say that I knew what the variant was. Could I go out there into these public knowledge bases and learn more about the variant? Could I explore it on my own? I don't want to be, if you will, held hostage to the information that the lab tells me. I want to be able to go out there and research a particular variant on my own. And when we looked at, sickle cell anemia, when we looked at thalassemia, when we looked at some of these other examples, it was a little bit easier to go out and search public repositories for the specific variant because the variant could be precisely named using HGVS. And everywhere you look, whether it's within HL7 version 2 or FIRE or anywhere out there on any of the public repositories, the by far predominant method for identifying variants is HGVS syntax. There's another syntax that's used for representing variants. It's called ISCN, and that is a syntax that's a little bit more amenable to cytogenetics, and we'll come back to ISCN when we start talking about cytogenetics during the genetics testing session. But HGVS is by far the most commonly used standard way of expressing the actual variant. And the utility of that is that it's often the best key for searching databases. When you have a variant, if you can express that variant in HGVS syntax, you then enable yourself to go searching these public repositories for more information. And when we look at the simple variants from the last few weeks, it was very easy to come up with an HGVS syntax. And the fact is, if 5 of us went off and came up with our own HGVS syntax independently, chances are we'd all come up with the exact same syntax. But when we start looking at structural variants, particularly variants that have imprecise breakpoints or variants where the breakpoints vary amongst family members, things get much more difficult. In fact, for this particular mutation, for the intron 22 inversion mutation, I've yet to figure out exactly what the correct HGVS syntax is. Just for fun, the CDC posts a resource. And by the way, the URLs for all of the stuff I'm going to be talking about are all in the slide deck. So the CDC resource, the Leiden Open Variant Database resource, they're all in the notes in the slide deck. So the CDC posts a database of intron 22 inversions, and they use a couple of different HGVS syntaxes. The Leiden Open Variant Society uses a different syntax, and actually I was able to communicate with the ClinVar folks and with Clem McDonald and just for fun with the HGVS folks as well. And I got different recommendations for the HGVS expression for this particular variant. So I have close to 4 or 5 different variants, 4 or 5 different HGVS expressions for this particular variant. And in an email I got from Clem, he says, in general, it's not possible to match up multiple reports on variations with vaguely defined endpoints, meaning it's hecka hard to search public knowledge bases for these types of variants right now because there's no agreement on what the actual use of HGVS is. And of course, couple that with the fact that HGVS is evolving as a syntax to keep pace with next generation sequencing technology. So you have a lot of things running in parallel. So if I haven't confused you enough, and probably if I'm confusing you, it's because I just find this area challenging. It's structural variants with imprecise breakpoints are tricky, in part because coming up with an HGVS expression is tricky, and because you can't come up with a definitive agreed upon HGVS expression, that means there's no key to search public knowledge bases. So for instance, ClinVar, which is really the primary repository for clinically significant variants, there's actually no HGVS syntax for this particular variant. But if you search ClinVar by factor 8 inversion, you will find ClinVar 10150. So in this case, you couldn't search the public knowledge bases by the HGVS syntax. And there's, half a dozen or a dozen databases out there that store factor 8 variants, and not a one of them was searchable by HGVS. They all had to be searched by for instance, intron 22 mutation or things like that. So pretty tricky, but that's just the way it is right now. And I expect that things will be, this is really a moving target, and I expect that over the next few years, the HGVS expression for these structural variants will improve dramatically. So once we can find the variant in public knowledge bases, obviously the lab is going to tell us what the lab wants to tell us about the variant, but sometimes we want to learn a little bit more ourselves. In this case, ClinVar indicates for record 10150 that it's pathogenic. And here's another case where there's no link to the SNOMED code. There's no link from ClinVar to the SNOMED code. And so here we have to do manual lookup to find that this represents factor VIII deficiency. And, in the past, we've been looking at the HL7 representation of the variants, but here what I'd rather do is focus on the family history representation, only because what we really want to be talking about in this session is inheritance patterns. So for here, we have a pedigree, and we want to figure out how do you communicate the pedigree. And we know that the proband, the patient, ST, has an unknown carrier state. We know that the brother has hemophilia. We know that the mother is presumed to be a hemophilia carrier. Here, you could say we've assumed it, but the father, he would manifest the disease. So I think we're safe to say he just doesn't have hemophilia. He hasn't been genetically tested, but he doesn't have any obvious phenotype. So we can just say he doesn't have hemophilia. And we know that the maternal uncle died of hemophilia. And so, let's see here. Again, you can grab this from the website. This is just a representation of in FIRE. FIRE has a couple of family history resources. There's the regular family member history resource, and then there's the genetics profile. This one does not use the genetics profile, only because I didn't need to. The real difference between the complexity in the profiles is whether or not you can express relationships with a coded vocabulary or whether you need pointers. So for instance, I can say that this person's my mom just by having a semantic relationship between me and this person as natural mother. And I can say that this person is my maternal grandmother or my maternal uncle because there are relationship codes for maternal uncle or maternal grandmother. But it gets a little bit difficult to express relationships when you're talking about, for instance, I have 3 kids and each of my kids have 3 kids, and I want to talk about a particular grandchild, but I need to say I'm talking about the grandchild that's the child of this kid. And so sometimes when we're building a pedigree, it becomes very challenging to express the relationship between people using just coded terminology. We need to use pointers. And one of the differences between the FIRE family member history profile and the family member history genetic profile is that the latter, the genetic profile, allows you to point. It doesn't limit you to just using coded terminology. The other main difference between the normal resource, the family member history resource, and the genetic resource is that the latter, the genetic resource, also allows you to include genetic observations on family members. So this is a fairly simple pedigree. The other thing about the resource, either one of the resources, the family member history resource or the family history genetic resource, is that I'm trying to say that the mother is a presumptive carrier of hemophilia, and I'm trying to say that for the father, hemophilia is not present. So I'm trying to negate an observation, or I'm trying to say that an observation is presumptive. And neither of the resources provide any fields for doing that, but both resources allow for SNOMED post-coordinated expression. And so, for instance, here, I wanted to say that the mother is a presumptive carrier. I wound up using a SNOMED expression of a finding with explicit context that the finding of hemophilia carrier is probably present. And likewise, for the father, I wanted to assert that hemophilia was known absent. And so again, I used a post-coordinated expression of clinical finding absent with an associated finding of hemophilia. And anyhow, and maybe that's cleaner, or maybe that's more complicated. I don't know what the state is out there of post-coordinated expressions, but at least using these resources, I couldn't figure out any other way to represent presumptive or known absence without creating SNOMED expressions. So here's the FIRE resource. The FIRE resource is it's a list where you list the individual family members using family member history resources. And then at the tail end of the list, you just say that this is a list of history family member diseases, and you reference the list items. The other thing is this is ST's family history. This one is generated from this is the My Family Health portal. And so if you want, you can take this XML. This is in line with the HL7 version 3 pedigree standard. You could take this XML, you could import it into that website, and you can look at the pedigree that I created. Or you can also go ahead and generate your own. Okay. So, any questions about the bioinformatics aspects of hemophilia?
Bob, and that that FIRE representation, the pointing, what is, and you have the family member 1 or family member 2. That what is? So you have history of family member disease, and then is this pointing somewhere else, like in this message, or pointing? What is it pointing to?
So the pointing between family members is supported by the genetic profile, but not in the base FIRE resource. This is the base resource, so there's no pointing in here. If I were using the genetic profile of this resource, basically it allows you to point from one family member history to another family member history, where the semantics of the pointer are either you're pointing to a parent or you're pointing to a sibling. So for instance, here I could point to, well, I don't think I have any, but let's say that I had a family member history here of grandma. Okay, let's say that this was grandma. Here, when I did the family member history for mom, I could say natural. I could have, for grandma, I could have used a relationship code that said maternal grandmother, but also down here, when I'm talking about mom, I could have said mom's mother is this one up here. So the pointer would have allowed me to say maternal grandmother as a way of saying it separate from what the coded terminology would have allowed me to say. So you can point from one family member history to another family history. You can reference another one where the reference says I'm pointing to the parent, or you can point from one family member history to another family member history where the semantics of the pointer is I'm pointing to a sibling.
Okay. So is it in the same message, or can you? Yeah. Like, would it have to be in the...
Sorry, say it again?
I was just asking, so if this would be a single message that you would be able to point to within that same message, or somehow you're pointing outside?
Yeah. Either way, because
Okay.
each one of these resources is identified. And so here, for instance, I'm asserting a list, and the list is just referencing, down here, the list is just referencing the members. Now, so here, this could just be one big resource where here within this family member history, I just include a pointer and points up to this one. This one could be in the same message, or it's not necessarily. So long as the receiving system could resolve the identifier, it would still be okay.
Okay, thank you.
Let's see, any other questions about this? This HEVS stuff is just super interesting, and that's the fact that nobody really knew the answer. In some ways made me feel better, because it means, okay, this is a good time for us to really dig into HGVS and begin to understand this stuff. We're a little bit ahead of the curve still by trying to really understand this stuff. And I really do think that HGVS is going to represent a key for searching these databases. For simple variants, it's without a doubt the best key to search all of these different databases, public databases. Unfortunately, it's not quite as reliable a search key when we're talking about large structural variants yet, but hopefully that will change, because what we'd like to enable, down the road is, you want to be able to just click and have access to this wealth of genetics knowledge out there in all these public repositories. And I think HGVS is going to give us that key, but we can see for some of these more large structural variants, it's just not quite ripe yet, even though it's very good.
Hey, Bob, this is Andy. Could I ask a question about, the example you're using, actually I think it was hemophilia B, but there's a pretty good correlation in terms of penetrance between the genome and the phenotype. SNOMED seems very well designed for coding or capturing, family history as, phenotypic disease. For things that have partial penetrance, or that are quote unquote carrier states, but of how do we actually transmit the family history of the genetic information, I suppose, beyond this sort of phenotypic information? Are you going to get into that later?
Kind of, sort of. I mean, I haven't taken so far, most of the examples, actually, so far, all of the examples I've wanted to build, there's different SNOMED codes for carrier versus have disease. So if you look at sickle cell, there's sickle cell carrier and there's sickle cell disease. And if you look at hemophilia, same thing. But when you think about all of the different diseases out there, it's hard to imagine that we're going to find all of the necessary pre-coordinated SNOMED codes. And what the HL7 messages do is they have a field for the variant, and then they have a separate field that says, is it heterozygote or homozygote? And the lab, depending on the method, the lab will make that determination. If they're doing next generation sequencing, they'll see that all of the reads at a particular spot, are an A, or they'll see that half the spots are an A and half the spots are a G, which will then imply heterozygote. So the way that HL7 and the genetics community is dealing with it is this a mosaic? Is this a heterozygote? Is this a homozygote? As just an orthogonal field. Right now in SNOMED, in the SNOMED model, there's no way to post-coordinate that. There's no expression semantics for post-coordinating that. And so I think we're left with right now just using the separate fields in the message, or in the case of this particular pedigree here, I was able to find, hemophilia carrier that I used for mom versus hemophilia, which I used for for brother and for uncle.
And you could have specified down to like carrier of A, hemophilia A or B, which would then imply, I presume, the specific gene product.
Right. And in the message, in the HL7 message, I could have been very precise. This is, in fact, I could have you can even include now the HGVS syntax in the message, and you can say, was it homozygote, was it heterozygote? You can also point to the ClinVar record. You can point to the pathogenicity. And so you can be very precise in the HL7 message where you have a lot of different fields, but you can't at this point, and I don't know if that's ever going to be possible, you can't bundle all that up right now into a single pre-coordinated SNOMED code. Does that kind of answer your question?
Yep, thanks.
Mm-hmm. So, and just a little bit about treatment. And we'll get into treatment quite a bit more down the road, but actually one of the earliest successes with recombinant gene therapy, where you actually take the gene, the human gene, and you put it into a bacteria, and that bacteria then winds up producing human factor VIII and factor IX. This is one of the earliest successes with recombinant gene therapy. And so we've been using human recombinant factor VIII and IX for quite some time. We'll actually talk a little bit more about what that means and how recombinant gene therapy works when we talk about genetic engineering. And we'll also talk more about gene therapy in a little bit more detail when we talk about genetic engineering. But here, for instance, there's been successful treatments of hemophilia B. You have an adenovirus that has had the factor IX gene inserted into the virus. The virus is then systemically administered to a patient. That virus then infects cells. So, for instance, it would infect liver cells, and liver cells live a long time. And there's cases where patients have been cured of hemophilia for years and years at this point, following systemic administration of these genes versus viral vectors. There's continuing studies ongoing. Some of the earlier studies, the virus wound up triggering cancer because it happened to insert in the promoter region of an oncogene. And so this stuff is always done with a great deal of caution, but there's also considerable optimism. So we'll be talking quite a bit about gene therapy when we talk about genetic engineering.
Yeah, so Bob, do you remember in the early 80s, before the recombinant factor therapy was available, and how many hemophiliacs were sort of, when we were first diagnosing AIDS, how many were coming down with the disease because transfusion was the only therapy?
Yeah, I recall it.
I'm talking about way back before we were able to test blood for HIV and things like that. Well, they were sort of like the canary. They were sort of like the hemophiliacs were kind of the canary in the mine for AIDS.
Yeah, I think it was as many as 50% of the hemophiliacs in the U.S. had HIV in the 80s.
Right.
And the lifespan from hemophilia was cut dramatically down to young 20s. Mm-hmm. Back then, yeah, it was.
But wasn't that actually true due to the fact that it was the replacement wasn't recombinant, but was actually collected from 100, 1,000 blood donors to a single hemophiliac?
Exactly. The recombinant therapy was not available then.
Yeah.
Right. Yeah, and we didn't know to sterilize the materials either for HIV. Now you can even give derived concentrates, but they can be treated to kill the HIV. But in the early 80s, we couldn't do that. So there was, now we have recombinant, which we'll use, and we'll have gene therapy. So now that obviously people with hemophilia are living much longer lives.
Yeah, I was working blood bank and microbiology in those days, so the cases were really dramatic.
Yeah. Okay. So just briefly, we're now going to be switching to talk about family trees. And this slide has far more, I mean, this is a science into itself. People go nutso with family tree representation. So I just kind of stuck this in there so you have a reference if you ever do see some complex family trees. We're going to be looking at family trees that are far more basic than this. But at least you kind of have this handy if you need it. And the next few slides are going to be a little bit of a review for people. First, we're going to talk about Mendelian inheritance, and then we're going to talk about non-Mendelian inheritance. And in Mendelian inheritance, Mendel actually had 3 laws, if you will. And when you see inheritance that follows Mendel's 3 laws, then it's Mendelian inheritance. And when it diverges from those laws, then it's non-Mendelian inheritance. And one law is that you have the homologous chromosomes here. Mom has 1 chromosome she got from her mom and 1 chromosome that she got from her dad. And one's going to go in one zygote, or one's going to go in one egg, and the other one's going to go in another egg. So you're going to have segregation. And if this is chromosome 7 and here's chromosome 5, chromosome 7s are going to split independently of what's going to happen with chromosome 5 or chromosome 3. So if the chromosome mom got from mom goes to this egg, well, for chromosome 3, that's not going to influence whether the chromosome mom got from mom or got from dad is going to go over. So there's the segregation of the homologous chromosomes. There's independent assortment, so that this pair has no influence over the other pair. And then the third law of Mendelian inheritance is the law of dominance. And the law of dominance states that at a particular locus or a particular gene, some alleles are dominant while others are recessive. Okay? And so here you see at this particular gene, there's allele A, and the capital A signifies that it's dominant. And at allele B, at this other allele, we have the little a, and that's recessive. And let's say dad has an A, a big A, and a little a. Possible offsprings are you can get 2 big A's, a big A and a little a, and 2 little A's. And again, if both chromosomes have the same allele, that's called homozygous. And if both chromosomes have different alleles, that's called a heterozygote state. Okay? So let's take a look at, one of the, probably better known Mendelian inheritance patterns called autosomal dominant. And so in this case, let's say mom has 2 little a's, 2 recessive alleles, and dad has the dominant allele and the recessive allele. And in an autosomal dominant condition, all you need is 1 allele in order to manifest. Or in other words, the phenotype is seen in the heterozygote. And what you can see here is that dad can transmit that gene to his offspring. And if mom has the condition, she can also transmit the condition to her offspring. You would expect to see the phenotype in every generation because if not, because it's dominant. And so if you see a generation where there's no phenotype, where no one has the disease, you assume that that mutant allele must have been removed, is no longer present. So in an autosomal dominant pedigree, you'll see every generation has the phenotype, and also we'll see that the child of an affected person has a 50% risk of inheritance. So here, this is a little Punnett square. You guys probably remember from high school biology, where if the affected person has a big A and a little a, and an unaffected person has a little a and a little a, then possible offsprings are the big A and the little a, and etc. So there's 50% chance that the offspring of the mating between an affected parent and an unaffected parent, there's a 50% chance that the offspring will be affected and a 50% chance that they won't. If we look at autosomal recessive, here the phenotype is only seen in the homozygote. For rare diseases, oftentimes you'll see what's called consanguinity, or mating between family members, between close family members, and that's what this double line represents, a consanguineous breeding or mating, or whatever the right word is for humans, having babies together. And in this case, this person's a carrier. They have the recessive mutation, and then they have the normal upper A, and then this person's a carrier. And if we look at our Punnett square, where you have both parents are carriers, you would see that in 25% of the cases, you would expect the offspring to have both of the recessive genes, and they would therefore be homozygote for that particular allele, and therefore manifest the phenotype. This is what you'd see, for instance, in sickle cell anemia that we've talked about. We're going to come back to this way early in the class. Several weeks ago, Rafiq asked a question, and Rafiq, we're finally going to come back and answer your question in a minute. And you may not remember what it was, but I'll remind you because it's a quiz question to see if you can get it now. Okay? So remember, when you have autosomal recessive and you have a carrier parents, the risk of having an affected child is what? 25%. Okay? So hold that thought, and then we'll get to the quiz question in a minute. X-linked is where you have the phenotype seen in males, and the females are carriers. Okay? And if the father is affected, then all of the daughters are going to be carriers and all of the sons are going to be normal. Okay? So if the father is affected, then all of the daughters are going to be carriers because all of the daughters get the father's X chromosome, but none of the sons will be affected because none of the sons get the father's X chromosome. On the other hand, if the father's normal and the mother is affected, then half of the daughters will become carriers and half of the sons will be affected. One of the clues here in looking at the pedigree is that you will never see direct male-to-male inheritance. Okay? You'll typically just see males affected, and you'll never see male-to-male inheritance. Inheritance will always have to go through a female carrier. Okay? Y-linked inheritance is a mutation on the Y chromosome, and therefore what you'd expect to see is all sons, and only the sons of an affected male, will have the mutation. Women won't have it ever, and 100% of the males will have it, assuming 100% penetrance. And an example of a condition that is represented by X-linked inheritance is hairy earlobes. So now you can see people with hairy earlobes and know a little bit about their genetics, maybe more than you wanted to know. Okay. Okay. So I jammed through that pretty quick, but I think you guys are probably familiar with that. But let's do a little quiz just to see. For this first pedigree here, what kind of inheritance is illustrated?
Y-linked.
Well,
no, wait a minute. Sorry.
Keep in mind it's got a female involved.
I didn't see that female. Okay.
Okay.
Yeah.
So you'll notice that you're seeing. Oh, well, let me give you guys a minute to ponder it before I say anything. You guys remember way back when there was this article in the Annals of Internal Medicine where they said, why bother examining the heart anymore with a stethoscope? Because now, heck, let's just do an echocardiogram. Is there still any value in physical exam? Well, luckily, the same cannot be said for pedigrees. And when we talk about genetic testing, one of the points I hope to make really clear is that by far, the single most important test in genetics is the family tree. Because that's how you know that a disease has a genetic component. So, any thoughts about this first one?
Autosomal dominant, I would say.
Yeah.
And why? Why would you say that?
Well, because we have males and females in that first generation inheriting it from the father at a high frequency, and female giving it to their sons, and the next one over, the mother and father have the disease, and they don't transmit it to everybody. Their kids, 2 out of the 3 kids have it.
Right.
And one doesn't have it. So that, I think the only way that could happen would be autosomal dominant.
All right. Autosomal dominant. Very good. Very good. How about this middle one?
I guess that is autosomal dominant.
Okay. So it's tricky, right? Because it's a small pedigree, and it could be autosomal dominant because you would expect in a pedigree 50% of the offspring to be affected, but there's a range, and so a woman could have 0, 1, or 2 of her children affected with an autosomal dominant disease. So this could be an autosomal dominant pedigree, and I probably should have given a bigger pedigree because that's not what I intended this one to be. And in fact, it's not what this one is, even though you're exactly right that it could be. But one of the things I would point out here is notice that, oh, you know what? We didn't even cover this yet. Oh, I'm going to have to fix this slide. Quiz. You know what? We didn't cover this inheritance pattern yet. It's coming up in the next slide. But we've covered it in a previous session. One of the things you'll notice is that the woman is transmitting the condition to 100% of her offspring. The woman is transmitting the condition to 100% of her offspring, and the man is transmitting the condition to 0% of his offspring. So can you think of any inheritance pattern where the woman
Mitochondrial DNA? Mitochondrial DNA related.
Yep. Yeah, that's exactly right. Murph, that's right. Exactly right. And my apologies, I shifted mitochondrial DNA because it's not really Mendelian, but there you go. Okay, how about this one? How about this one on the right? Let's see, what do we see here? We see...
Is it X-linked inheritance?
You have, well, you have an equal number of females and males affected, which doesn't completely rule it out, but it argues against X-linked because normally the women would be carriers.
Seems like it's autosomal recessive.
Autosomal recessive, and why do you say that?
Well, for parents have 100. If both parents are affected, 100% of their children are, and if one parent is affected, 25% of their siblings are, or their children are.
Right. So presumably, this person here is a carrier, and must have married someone else here who's also a carrier, and that's why they were able to have a child that had the disease. Right, so this would be an autosomal recessive pattern. Rafiq, I don't know if you remember asking this question way back when?
The first session.
The first session. So I thought it would be good now. We talked about the fact that when two carriers, so let's assume that these two people here, thanks Andy, when these two people here mate, right, they're both carriers, there's a 25% probability of having an affected child. But what if we don't know that these two people are carriers? What if we're not quite sure? So this cystic fibrosis is an autosomal recessive disease with a carrier frequency of 4%. So there's a 4% chance that mom is a carrier, and there's a 4% chance that dad is a carrier. In a random mating between two people, what then is the probability of having an offspring with cystic fibrosis? So how would you calculate that? So the answer, it's actually pretty straightforward. It's just you have 4% times 4% times 25%. Okay, so it's just 0.04 times 0.04 times 0.25, and you wind up with a probability of having an offspring with cystic fibrosis of being 0.04%. Okay, and that's the answer to your question from the first week, Rafiq. Okay.
So that's just in the general population? Is that what you mean?
That's right. That's right. In the general population, the carrier frequency of cystic fibrosis is 4%. So 4 out of 100 people just selected at random would actually have a cystic fibrosis heterozygous mutation. Because it's that common.
And what's the?
I mean,
Sorry, go ahead. Finish your thoughts.
Oh, I was going to say, 4%. That's really 4% is in Northern European descent. In other populations, it's nowhere near 4%. But this is really mainly just to illustrate the point. So what were you saying?
What's the probability if both parents are not carrier, but a still child gets some genome abnormalities, especially on the deletion side?
So if neither carrier, if neither parent is a carrier, the likelihood of them getting cystic fibrosis because it's autosomal recessive is astronomically small, because you'd have to have a mutation occur both in the mother and in the father. So that's astronomically small. It depends on the mutation rate, and I don't know what the mutation rate is for this, but if you're talking about an autosomal dominant disease, there oftentimes you will see, there's no parent that has the disease, but the child has the disease. See, that only requires that there be a mutation during gamete formation in one of the parents. And there it depends on what the particular mutation rate is for the particular disease in question. And the mutation rates, they range, some of them are as high as like 1 in 10,000 or 1 in 100,000 for more rare conditions. So again, if neither parent is affected, the likelihood of them having a child that manifests an autosomal recessive disease, I would do DNA testing on the postman, to tell you the truth. But but if it's an autosomal dominant disease, then it depends on the mutation rate for the gene in question. And that type of stuff is cataloged, and there's resources where you can look up mutation rates for particular diseases.
Okay. And I think the probability of having a deletion is higher if during the mutation, right?
I'm sorry?
So probability of deletion, a small segment, is higher than any other these kind of diseases, right? Autosomal disease.
Point mutations, if you're talking about new spontaneous mutations, point mutations by far are the most common.
Mm-hmm.
Let's see here. There was a table I showed you. See if I can pull this up real quick. From last week's. And if we look at this, you'll see that disease-causing mutations are almost always, is that readable? Let me see if I pull that up a little bit. This will give you a rough idea. Large deletions, inversions, fusions, duplications account for a relatively small percentage of new mutations. Most of the new mutations are actually nucleotide substitutions.
Yeah.
Okay.
Thank you, Bob.
Mm-hmm. All right. You ready for some non-Mendelian inheritance? Okay. All right. These ones are, well, boy, they really make you think. Now, this one is pretty straightforward. This is autosomal dominant inheritance with decreased penetrance. And there's a lot of autosomal dominant diseases that don't manifest 100% of the time. An example of this would be familial breast cancer due to mutations in the BRCA1 gene, where the likelihood of someone who's inherited that mutation, of developing breast cancer is about 80%, not 100%. So it's possible that even though it's an autosomal dominant inherited mutation, that you won't necessarily see breast cancer in every generation, and yet this person here still inherited the autosomal dominant gene and can pass it on. So this looks like autosomal dominant, but it has decreased penetrance. And so you may see that there's generations where there's an apparent skip in the phenotype. We talked a little bit in the past about
hey, Bob,
mitochondrial.
Excuse me. Bob, do you have a quick explanation for the basis of penetrance?
Well, it's probably, at least oftentimes, due to the fact that my other monitor. Oh, there we go. It's due to the fact that really, when we look at things like sickle cell disease, we try to simplify the world by saying, well, this is a monogenic disorder. But in reality, for almost everything we look at, there's this notion of modifier genes. Even in sickle cell disease, if you also happen to have a mutation that leads to what's called hereditary persistence of fetal hemoglobin, you may go your whole life without ever obviously manifesting sickle cell disease because you have fetal hemoglobin, and that does the job of decreasing sickling. So for the most part, well, at least in a lot of cases, decreased penetrance is because no disease is really the result of just one gene all onto itself. There's always modifiers. There's always environmental factors. And maybe this person wasn't exposed to gluten, or maybe this person never had a strep infection, so never got rheumatic heart disease, or maybe this person also had hereditary persistence of fetal hemoglobin on top of their sickle cell disease. So generally decreased penetrance, you can think of it as being because of the presence of modifier genes and or because of the presence of environmental factors that may or may not have been present.
Great. Thank you. That makes good sense.
Mm-hmm. We talked about mitochondrial inheritance, so just for completeness, I included it here, and we'll just take a quick look. But again, mom, especially for a homoplastic female where all of her mitochondrial DNA is mutated, then 100% of her offspring are going to show the phenotype, whereas males, there's no mitochondria in sperm, and so males will have 0 offspring that have the phenotype. Sometimes you see, now I'm going to get into a couple of rare conditions, but they're super fascinating, so I hope you guys will enjoy these. Sometimes you'll see these pedigrees that look, for all the world, like they're autosomal dominant, but with each generation, the disease, the phenotype manifests, the onset gets earlier with each generation. The phenomenon is called anticipation. So this looks, for all the world, like an autosomal dominant condition, and yet it manifests at a younger and younger age with each generation. This is a pedigree of a family with myotonic dystrophy, and here's a pedigree of a family with Huntington's disease. Huntington's chorea, which I think a lot of people have probably heard of Huntington's chorea. It's one of the devastating aspects of Huntington's disease is that you live, till you're 20 or 30 or 40 or 50. Only then do you start coming down with manifestations of the disease. So by then you've already had your kids, and so it was too late for genetic counseling to help you decide what to do. But Huntington's disease also manifests anticipation in that successive generations begin manifesting symptoms at a younger and younger age. So what might be going on to cause anticipation? What the heck is anticipation? What could be going on here to make the disease manifest differently in successive generations? And this slide tries to illustrate what's going on in anticipation by zooming in on Huntington's disease. Huntington's disease is a mutation in the HTT gene. This gene produces a protein called huntingtin. The exact function of the huntingtin gene isn't known, but it looks like it plays a critical role in brain development. If you look in a normal huntingtin gene, you'll see there's this long stretch of CAG repeats. Now, last week, when we talked about polymorphisms and we talked about DNA profiling, we said, there's a lot of places in the genome, maybe 10,000 different places, where you have these trinucleotide repeats where one person might have a string of, say, 7 repeats, somebody else will have 10 repeats, and this is one of those. It's a short tandem repeat where there's considerable polymorphism in humans, although this particular short tandem repeat happens to be in an exon, whereas most of the short tandem repeats occur in introns. So this particular short tandem repeat of CAG trinucleotides is actually within the exon. So every time the a number of CAGs increases, you wind up inserting another amino acid into the protein. And what happens is the longer the stretch of CAG repeats gets, the more unstable the region of the exon gets, and the more prone it gets to getting even longer. And that is what accounts for the anticipation. You have one generation that has, say, 5 repeats. The next generation might have 10 repeats, but now that there's 10 repeats, you're more prone to having additional repeats, and you get even more repeats. And as the number of repeats gets above 35, you have manifestations of Huntington's disease. And as the number of repeats increase, the age of onset of Huntington's disease begins to decrease. And so the anticipation that you see with these conditions is because of these repeating segments that as they get longer, the risk of them mutating even further goes up. And so with each generation, you see more and more mutations occurring, and the more mutations that occur, the worse the disease is and the earlier the age of onset. And that's why we see anticipation with Huntington's disease. Another interesting pedigree is illustrated here. This illustrates something called imprinting. Here we see that when the female has the disease, it looks like an autosomal dominant inheritance pattern. She gives it to about half of her kids. She can give it to males and she can give it to females. Whenever the female has the disease, okay, it looks like it's autosomal dominant. Okay. When the male has the disease, he's not passing it. None of the kids manifest. But the female's a carrier. Now the female's a carrier, but wait a minute. She's still looking like an autosomal dominant? Wait a minute. What's going on here? What's going on here? It looks as though only the maternally inherited allele is being transcribed. Okay, so this child here is inheriting a chromosome from dad and a chromosome from mom, but the chromosome from dad is being ignored. Only the chromosome from mom is being transcribed. This child here got a chromosome from dad and got a chromosome from mom, but happened by chance, by 50% chance, to get the diseased chromosome from mom. Here, dad has the disease, and dad transmits the diseased chromosome, but mom had a good chromosome. So this person here has the diseased chromosome from dad, but the normal chromosome from mom. And since only the maternally inherited allele is being transcribed, this person is a carrier, but doesn't manifest the disease. That's a mouthful. That's a mouthful. I'll show you a couple more pictures to better explain this. But in imprinting, what's going on is there are many genes, close to 100 genes we know of, where the child only uses the maternally inherited gene or only uses the paternally inherited gene. Okay? The other one is silenced. Here's a case of paraganglioma syndrome 1 caused by a mutation in the SDHD gene, and here only the paternally inherited allele is being transcribed. So mom had the disease, none of the kids manifest the disease, but they're all carriers. Where dad is the carrier, he can now transmit the disease, but where a female is a carrier, she can't transmit the disease. So this is kind of a crazy condition. It's called imprinting. What happens in imprinting is, okay, so dad is producing sperm. And when dad produces sperm, he marks key genes on the sperm within the chromosomes in the sperm. He marks key genes as these have come from dad. First, everything gets reset so that by the time dad produces the sperm, you can tell in the sperm which genes are length is coming from dad. And mom does the same thing in the egg. She flags certain genes as these are genes that come from mom. So now in the child who inherited this chromosome from mom and this chromosome from dad, dad flagged this gene as coming from dad, and mom flagged this gene as coming from mom, and some cells will respect those flags, and some cells will not be able to use this particular gene that came from dad because dad will have silenced it. Some cells will only be able to use the maternal copy of that gene, whereas some cells will also only be able to use the paternal copy of this other gene. And that's called imprinting. And there's about 100 genes that are known to be imprinted, and then there's a number of diseases associated with imprinting. So for instance, we have these diseases, Prader-Willi syndrome and Angelman syndrome. On chromosome 15, in this particular stretch, there's a region where we only use the segment of the chromosome that we got from dad. We never used that. We never used the segment that came from mom. Mom imprinted that region and silenced it. There's a separate area on chromosome 15 where we only use the segment that came from mom. We don't use the segment that came from dad because dad epigenetically silenced that region. He imprinted it. And so if dad has a deletion of this entire region, the Prader-Willi region and the Angelman region, the only part that matters is the Prader-Willi region, because the child is going to use the Angelman region from mom anyhow. But the child can't use the Prader-Willi region from mom, has to use the one from dad, and that was deleted. I know that's kind of confusing, but really what imprinting means is that you have 100 or so genes where dad will imprint some of them and mom will imprint some of them. And by imprinting a gene, it means that you've made that gene inactive. And so when the child inherits the gene, if dad imprinted a gene, the child can't use it. The child has to use the other copy of the gene that came from mom. And so that's why we get some of these unusual pedigrees because of imprinting. That's a mouthful. Any questions about that?
Is that imprinting ever reversible? I mean, it seems to me theoretically possible.
It only gets reversed when, like I inherit a chromosome from dad and I inherit a chromosome from mom. But when I now produce my own sperm, all of those imprinting marks get erased, and I now imprint regions, and then it starts to cycle over again. So the only time the imprinting gets erased is during oogenesis and spermatogenesis. But otherwise, throughout the life of the individual, the imprinting marks remain. And so there's a number of genes where you always only use the copy you got from mom, or you always only use the copy you got from dad.
Thanks.
Mm-hmm. Let's see. We're doing pretty good on time here. Mosaicism. Probably people are most familiar with mosaicism because of cancer. I think most people have now come to understand that cancer is due to mutations that occur in body cells or somatic cells as opposed to germ cells. So where you have these mutations occurring in somatic cells, you wind up having a population of cells that differ from the neighboring population of cells. One extreme example, we'll come back to mosaicism when we talk about cancer, but one extreme example of mosaicism, normal mosaicism, is X inactivation. And within the first week of embryogenesis, one or the other X chromosomes is chosen at random to become inactive. So in every cell of the human female, one of the X chromosomes is active and one of them is inactive. But in a neighboring cell, the X chromosome that's active or inactive can differ. There is an X inactivation center. It's a gene that codes for what's called it's an RNA. It codes for an RNA. That RNA doesn't produce a protein. It's called a long non-coding RNA. And so the gene, the X inactivation center gene, gets transcribed into a long non-coding RNA. You can kind of picture that long non-coding RNA wrapping the chromosome that's destined to be inactive. People have seen the long non-coding RNA staying in the nucleus in close association with the inactive X chromosome. The inactive X chromosome is visible as a bar body in the cell. And what you see here in this picture, for instance, is mosaicism in Duchenne's muscular dystrophy. This is a cross-section of a muscle stained for the dystrophin protein. And the dystrophin protein is right underneath the plasma membrane of the muscle fibers. And here, in a normal female, you see the dystrophin protein. And here, in a male that's afflicted with Duchenne muscular dystrophy, you see the absence of dystrophin. Here's a normal female carrier where she's phenotypically normal, but because of X, and by the way, the Duchenne muscular dystrophy, the mutation is a mutation in the dystrophin gene, which is on the X chromosome. And so here, where you see a carrier, a female carrier of Duchenne muscular dystrophy, even though she's phenotypically normal, you can see that some of her muscle cells have inactivated the normal X chromosome and therefore have the absence of dystrophin gene, whereas other muscle cells have, just by random chance, inactivated the X chromosome that carries the DMD mutation, and therefore it's transcribing the normal DMD gene, and it looks like a normal muscle cell. So you have these normal muscle cells, and you have with complete normally dystrophin, and then you have these other ones where it's completely inactive because of the silencing of the X chromosomes in those cells. Okay, a few more. If you guys can bear with me, we got a couple more types of non-Mendelian inheritance, then we're going to do a quiz, and then we're going to wrap up. You got a little energy left? Okay.
Sure.
Cool. Go,
okay.
Bob, go.
All right. Thank you.
I'll be your cheerleader.
Thank you, Diane.
You need it. Okay.
Do you remember when we talked about epigenetics a couple weeks ago? For instance, we talked about how the promoter region of a gene might be methylated, and how methylation of the promoter region of a gene might change the amount to which that gene is transcribed. It turns out that these epigenetic changes, some of them are also heritable. So it's not just as we used to think. It's not just that changes in the actual DNA sequence are the only things that lead to heritable changes. It turns out, it appears as though epigenetic changes such as methylation of promoter regions can also be inherited. In the lab, in animal studies, we've seen epigenetic changes inherited across multiple generations. In humans, there's evidence for at least several generations of epigenetic inheritance. That means if you are exposed to, for instance, some event, some horrific event, and that horrific event triggers epigenetic modifications of your genes, there might still be physiologic manifestations of that horrific event in your grandchildren, because these events can alter your epigenetic profile, and the epigenetic profile can be then transmitted. So it kind of goes against the Darwinian notion of inheritance, because it means that things that happen to you can alter your epigenetics, and those epigenetic marks can actually be inherited. It's different than what we talked about with imprinting. Imprint marks get reset during spermatogenesis and oogenesis, but it appears as though other epigenetic marks don't get reset and in fact are heritable. Finally, I want to talk to you a little bit about multifactorial inheritance. And by far, this is the most common type of genetic disorder. These are the disorders like diabetes, like hypertension, like autism, like schizophrenia, where, you see a little bit more frequently in a family than you'd expect to see, and yet the pedigree doesn't show a classic Mendelian pattern. So you look at this family tree here, and you say, well, what kind of inheritance pattern is this? Well, it doesn't neatly fit into any of the pedigrees we've talked about. It's not autosomal dominant, recessive, X-linked, et cetera, et cetera, et cetera. And yet, it does seem to be that there's a little bit more of this disease in this family than you'd expect to see in the population. And this is the type of pedigree that you're going to see with things like diabetes, hypertension. You're going to see familial clustering without a clear Mendelian inheritance pattern. And what this usually signifies is there's multiple genes involved, and or there's nature-nurture. There's a genetic factor, but there's also non-genetic factors at play. And early on, in an earlier session, we looked at this picture here, and I just thought I would throw it in again to emphasize the fact that, sickle cell disease is a nice way of illustrating the central dogma of biology, but it's also somewhat misleading in that it can lead you to believe, oh, you have a gene, it's mutated, leads to a genotype, which leads to a phenotype. But by far, most genetic diseases are polygenic or multifactorial. Multiple genes are involved. Environmental factors are involved as well. Let's see here. Okay. So when we look at these multifactorial diseases, you can see in this table here how much more common they are than disorders due to chromosome mutations. These disorders due to genome and chromosome mutations would be large structural changes. They would be like trisomy, or they would be large segmental replications. Disorders due to single gene mutations would be things like sickle cell disease, but by far, most genetic diseases are multifactorial. They're things like diabetes, they're like hypertension, where we know that there's genetics at play, but we don't know all of the genes that play into a disease, and we don't know the complete mix of environmental factors and genetic factors. In fact, we're going to come back to multifactorial inheritance next week when we really begin to delve into diabetes, type 1 diabetes, and how genetics leads to autoimmune disease. When you see these pedigrees and you look at them, you say, gosh, you had cancer and your brother had cancer? Well, I don't know. I mean, is that just random chance? Or, maybe there is something going on there. How do we tease that apart? How do we begin to tease apart whether or not there actually is a genetic factor there? And we'll come back to this again next week as well when we begin to explore how the heck do we actually figure out what gene did cause a disease? How do we figure out that a disease is genetic in the first place? And from there, how do we actually figure out what the genes were? But here, what you see for these diseases that don't have clearly Mendelian inheritance patterns is we can do risk ratios. And you'll see that the risk of, for instance, schizophrenia is the risk ratio between siblings is 12 to 1 compared to the risk ratios between two random people. So if I have schizophrenia, the risk of my brother getting schizophrenia is 12-fold higher than it would be if he was just some normal person from the population. And so risk ratios are often used to determine that a disease has a genetic component. And the other thing that's often used is concordance rates in monozygotic and dizygotic twins. If you see a disease that has a much higher concordance rate in monozygotic twins than dizygotic twins, that's a clue that the particular disease might have a genetic basis, not a complete genetic etiology, but a genetic basis.
Yeah.
And this picture here kind of summarizes characteristics of complex diseases. Again, we're going to come back to this when we talk about diabetes because it is a multifactorial complex disease, and we're going to look specifically at how the genetics of diabetes was teased